So for our final review, if I would put it all together, you need to be able to explain how we get from normal cells to sickle cells from one little point mutation. So first, let's look at the point mutation. So sickle cell is a huge long DNA sequence, but right here in one single point, we change an A to the T, which then changes our mRNA. Instead of our mRNA being GAG, it is now GUG. Alright, which then changes glue, which remember is hydrophilic, to valine, which is hydrophobic. And that causes these to stick together where the valine and the hydrophobic properties are. Whereas hydrophilic, these guys would all separate. Because remember, hydrophilic or polar, they like each other. They're polar, they do not like hydrophobic things, so that hydrophobic pocket is going to repel each other, and they separate in the red blood cell. In sickle cell, they form rods. Alright, so if I would just go to just a blank slide here, basically, again, if we have hemoglobin, we have this hydrophobic pocket on the outside. And in normal sickle cell, this glue, tamic acid, is hydrophilic, so it's going to repel. Alright, because this is polar, this is non-polar, this likes water, this one doesn't. And glutamic acid likes water, this one hates water, so they repel each other, so this is our normal round cell. Whereas in sickle, we have that same hydrophobic pocket, and I'm not going to write hydrophobic again, but I'll just signify it there, but now we have valine, which is also hydrophilic, so that allows another hemoglobin to come in and stick together. And then another one. And another one, and so on and so forth, until we get these long rods, which when we try to fit them in a round cell, those straight rods cause it to be misshapen. Alright, and we get this sickle cell, and it's very stiff, it's not flexible. This one's flexible, it can go through our vessels right. This one is inflexible and gets stuck. So there you go, that's sickle cell. So that one single base changed this hydrophilic amino acid to valine, a hydrophobic amino acid. So instead of repelling each other, they connect, causing rods in the cell and it being inflexible and getting stuck in your blood vessels. And that is sickle cell.